Give me 1 second. Good morning. Can you see my slides? Hello? Can you hear me?
Yes, yeah,
Okay.
can hear you. You can see your slides.
Okay, thank you. I'm not sure if you want to do an introduction or something. I'm waiting a bit, so sorry. Okay.
Daniel, you want to, sorry, Daniel, you want to do the introduction, is it?
Hello, can you all hear me?
Yeah,
Hi, yeah.
good morning.
Yes, hello, Joachim, hello, Nicole. So we're very happy to conduct this event together with OLS and Gaia to learn more about 3D culture of iPSC organoids and in particular the CERO 3D Incubator and Bioreactor. So Bunseng is unable to make it to this event, unfortunately, but we have other representatives from IMCB, and we look forward to learning more about this and hopefully partnering with you all in the future. So yeah, that's my introduction. Please proceed.
Okay, thank you very much.
Thank you.
Thank you very much for stepping in. And just for all, normally I like this presentations to be a bit more interactive. So if you have any questions, just step in. If you want to focus me on some things or more particular or something like that, do not hesitate to interrupt me or just to raise your hand and come up with questions. Good. So until I don't hear anything, I will just continue. And thank you very much for the invitation and the opportunity to introduce this solution to you. Actually, just a quick overview where we are, who we are. We are from Germany, based in Germany, and we are a family business since 2004. And we're a bit in both worlds. So we are a distributor, but we're also a manufacturer. And here we're talking about the products that we produce in Germany and then are able to sell, obviously, globally. Just to give you a bit background, so we're quite familiar with the cell culture market. So we carry like imagers, we carry like incubators, flow cytometers, cell line development instruments, and so on. So we have a whole bunch of instruments and solutions and consumables in that field. And these are the solutions we have globally. A lot around, especially tissue culture, so tissue dissociation without enzyme. Quickly shows 2-3 slides about the CASY. Then zero plates, which are extremely good for imaging because they're relatively scratch-free. And also work nicely with fluorescent. And then obviously the major part will be about the CERO itself. Quickly, the tissue grinder, I'm going to talk about it 2 minutes only. So it has 4 slots. It's quite quick. So it's purely mechanical, and that works a bit like this. So you have a rotator, you put in the slides, you have a stator, and this is a cell strainer. Once you put that together, you can start the protocol. It takes usually 2 to 5 minutes. And then you can turn it around, centrifuge it, and then you have the cells pass through the strainer in the bottom. Just quickly, this is a mechanism. So we have teeth inside, so it's a sharp end and a blunt end. And with that bidirectional rotation, we first cut the samples and later on we grind them. And then after centrifugation through the strainer, you have the single cells on the bottom. If you have more questions, as I will not touch much more about it, let me know, let Gaia know, and we can go in more details. What is more central is obviously cell counting, and I know many people don't pay much attention to it, but actually now we are looking very much into a lot of literature, also research, and in many good publications, standardization is a key process. And standardization means reproducibility. Let it be that you have 3 different cell types you want to compare and or other things, then obviously a robust result with a CV, standard deviation of 2% or 1% or lower is probably very critical to have comparable results and to keep the number of repeats down. We can talk about that in more detail if you're interested. The next thing is, and that is something which is very strong, which comes with our system, is a volume detection. I think we have the best volume detection you can get currently. And I will tell you later in the next slide how that works. That is obviously of significant interest if you work in immunology, like T-cell activation, but also sometimes monocytes development, but also in the field of, sometimes we come to the next point, iPSCs and aggregation. Sometimes you see already differentiation, unwanted or wanted, when the cells try to drift, and or toxicology in experiments. Aggregation detection and heterogeneous cells, PBMCs, is one of the strengths, especially and very often used also in vaccine development and PBMC-related work. How does it work? Very simply, we have actually optimized the Coulter principle. So Coulter basically says we have two electrodes, we have a pore, cells pass a pore, the passage gives a pulse. So that's so far so easy. Now, the difference in what we do is we don't go for the amplitude to decide how big a cell is, because that has some risk. Like, I try to simulate this with the elongated cell. If an elongated cell passes a pore like this, then what you get is a very sharp peak. So very narrow, very high. On looking at the amplitude, this would look like a big cell. Now, if you just in your mind turn this 90 degrees around and you passage the cells like that, then you would get a long peak, but a flat one. So Coulter would say, the first one was a big cell, the second one was a small cell, but obviously they're both the same. So what do? We just scan with 1 megahertz during the passage and look at the pulse area. And that is in both scenarios a cell, the same. And that makes it very strong, like if you go look at neuronal stem cells, or if you look at all different kind of cells with a shape. Worst case, we have some customers doing bacteria, like Leptospira, things like that. So that works quite precisely. And this is extremely also valuable then, because you get a very precise volume of the cell. And that is then helpful if you, like in your case, maybe work with iPSCs. iPSCs very often are not passage as single cells, but small aggregates, or you have a lot of aggregation. And to analyze them, you may have to singularize them with a pipette or something. Very often what we see is that the results are terrible, because the killing rate during stimulation is different, and that's all some things we don't have to do. We can analyze the volume of an aggregate very quite simple. We know the cell volume of a small cell, of a single cell, and then it's pure math to divide the volume of the aggregate by the volume of the single cell. Here's an example of a cell, which we have the size distribution in brown in the back. And you see there's a majority around where the green arrow is, but then it goes down like, it doesn't drop down quickly in a Gaussian style, but it bleeds out a bit. And that means there is aggregation. And there's even a bit more in this area. And you can say like, yeah, but the majority are single cells. Yeah, that's true. But if you look at the cell volume, so the total biomass, you see that in the peak area, there's not much volume. The majority is in aggregation. And this is something we very often see also for iPSCs. You look at this because this is what you can count, but you ignore that, which makes up the majority. And that can end up with lack of standardization or very high deviations in cell numbers. Just one example. And as said, we can do a lot, from bacteria to platelets to yeast to blood parasites, up to all different kind of cell types, primary cells, cell lines, immune cells, and so on. Good. Let's park the CASY here. Quickly, just if you're doing imaging in plates, we have 24 well plates and 6 well plates with a flat button. We have, which goes quite well, the 96 and 384 as a U button. Ultra low attachment plates, and one of the strengths we hear from our customers is that they're very resistant to scratches, which makes it, and together with capability or compatibility with fluorescence, makes them a quite nice tool for imaging and high throughput imaging. So 2 examples just here. Good. I hope I didn't lose you on the quick summary. Now we go into the main part. You still there?
Yes,
Okay.
we are still here.
Okay.
Yeah.
Good. So this is a CERO we're going to majority talk about today. It's a standalone incubator. It has a bit the size of an inkjet printer for the older generation. Sorry for saying that. It's quite compact, and it can carry up to 4 tubes, as you can see here. And if you hope you can see my cursor. And what we do is we don't spin; we do bidirectional rotation. So we go one way, we stop, we go the opposite way. What happens is we have this, you see them here, small fins on the inside of the tube. And with that, we keep bigger structures, cells or bigger structures, free floating. So it's bidirectional rotation. We have 4 tubes. This chamber is controlled for CO2 and temperature. So it's a full incubator system. The tubes can be operated independently. So we can have 1 tube, up to 4 tubes, controlled independently for experiments. And this bidirectional rotation, I think the major strength there is that we can generate an environment where they are free floating and very stress-free. And that very often what we see and together with Metrogel-free incubation supports that the cells are developing faster, differentiation more quantitative. So very often, like with cardiomyocytes, I'll show you later, have 100% differentiation, more maturity, and so on. Good. I can tell a lot, but maybe I show you some data soon. One of the things, and I come to this, is just keep that in mind, the media change. If you want to do like 10 million cells or whatever you want to do, or 80,000 organoids, you know how many wells you have to process every week or every day, every second day, and how much time that costs, how much material that costs. Here, we just take a tube, we bring it to the hood, you aspirate, you add, you bring it back. So media change usually takes not more than 2 to 5 minutes. So it's quite quick. The time required depends a bit on how much excess volume you take on the media. And we have some groups that, like with kidney organoids, they go up to 80,000 organoids in 1 tube in 40 ml. It's roughly 500 micrometers. So you can see there is even a very high capacity in one of these reactors, which are quite flexible. You can use them from, worst case, 1 to 3 milliliters for short term, or in the long run, 10 to 50 milliliters. So it's upscalable. What are the main applications? I usually have 2 pockets. One is those working with pluripotent stem cells, iPSCs majorly, iPSCs and embryonic stem cells, not pluripotent, but stem cells, MSCs, and also their differentiation then into different kind of stem cells. So it can be even a seamless process. And then on the other side, we have groups that work with tissue culture. So adult stem cells, there are many names for it, cancer or tumoroids, whatever you want to call it. It's a slightly different protocol, but in the same machine. And obviously, both go there, but majorly here, we have also a lot of applications which go into co-culture. So either you have healthy and diseased tissue, or you have tissue first cell type, like one group is doing endometrial. I think they had stroma cells, and then they had T cells, to see how the immune reaction is. Or we have later on one example of a group that does microglia and brain, so different kind of applications. And the advantage is it's all without the need to use any kind of hydrogel as a kind of structural protection or something like that. It's just cells floating around. We use sometimes Metrogel, but I come to this later, but very rarely. Obviously, we can do long-term cultivation. We were just talking yesterday with a group that was doing brain organoids, and they were growing them up to 7 millimeters for some testing, and cannot tell more about it, but you can see even you can grow them up to 4, 5, 6, 7 millimeters. and the risk of apoptosis is significantly reduced in the system. As you can imagine, the organoids, the spheroids, are free-floating. They have perfect supply of nutrition, and also at the same time, metabolites are easily washed away. So these are, I would say, the main applications, and then we have some niches. Niches means where, for example, one group that has now 3 publications around, they produce CAR macrophages for testing in high amounts in the CERO in a continuous process. So they grow the CAR macrophages, and they can harvest them once a week over a period of several weeks. So if you're interested, we can share some publications about it. And then we have some groups that want to maintain tissue, so that also works quite well. One example would be, normally if you work with liver tissue and you get a quick fresh dissection, and you have them in a static model or something like that, then you have to process it within roughly 1 day. Otherwise, the tissue goes down. We have results that show that with human or liver tissue, if you cut it into pieces of, let's say, around 1.5 millimeters, you can keep it up to 27 days intact. So there's and that also supports that we keep provide an environment in the system that is very positive for keeping tissue or developing tissue. Any questions at this stage? I cannot see if somebody raises a hand. You have to raise your voice, unfortunately. Okay, no question. So this was getting from a presentation from Clevers, because it makes a nice introduction into the first part of work with organoids, which is derived from pluripotent stem cells. And this is probably something if you work with iPSCs, you all know. So you have to expand them, and I wanted to do the math. You start with the 6 well plates. You probably have 12 mm of media in there. You start with 800,000 cells in the per plate. You have 6 interactions. It takes you 15 minutes in one plate. Then you have to process it. You go to 24, 60, and so on. And I just done different splitting rates here on the bottom. And target is to get around 16 million, which is just in the middle between here. And it takes you 8 days, and it takes you 180 milliliters of the relatively expensive media. It requires you plates with matrigel and so on. And it's quite labor intensive, right? So I have just 20 euro per hour labor. I think that is not a realistic value for Singapore. But okay, so this value is probably even higher. But to give you one idea, your cost will probably be between 10 and 16 euros per 1 million cells. This would normally drive it even much higher. If we go with 50 or even more, then it's quite high. What you always have to do with the package, you have to check if there is some spontaneous differentiation. You need matrigel, which is not 3R, and you have to get rid of the matrigel, and the cells get used to it, and it's very labor intensive. But you know that, right? So what is the alternative? Expansion in CERO. So if you expand in CERO, there's a standard protocol. You just take 10 ml of media, and you add roughly 800,000 cells. And then you just add 5 ml of media for a sequence of normally 5 days. To make it more, let's say, I took the best scenario for the iPSCs, and I took the worst scenario for the plating. I took the worst scenario for the CEROs. You take 7 days. Normally, as said, 4 to 5 days you are there now with a normal expansion rate. But let's say on 7 days you have to add. That means you drive the media cost down 75%. You drive the cost for plastic down by 32%. You drive the labor cost down by 84%. Okay, even if you don't drive the cost down, the people can do other things, right? You can do more creative than just working on the hood. So if you look at it overall, you get down the cost 75%, plus you have some advantages coming with it. Advantages would be that, for example, you can have direct embryonic bodies, which you can differentiate. That's one. Another thing is what we normally don't see is any kind of drifts or unwanted differentiation. That's something which we don't see. And the differentiation which can come next is has been done in a lot of tissues like hepato, neuro, cardio, kidney, retina, and probably more. It's a seamless process, and you see there are the organoids. This is a kidney organoids. You see the nice polarity, nice differentiation that was done in Cologne, and no matrigel through the whole process. So it's a big advantage also that we have no need for matrigel. Let's quickly look at some results, published data. So this is normally from a plate. You put them in here, they form automatically the organoids, the spheroids, and they grow as a spheroid. This was published already in 2015 with a prototype from Life and Brain in Bonn. And you see they did the passage over 10 passages. And then after 10 passages, they looked for, and with immunostaining, that the cells maintain their pluripotency on the one side, but also can be differentiated into all three germ layer on the other side. And as said, no matrigel. Some other data here from the Max Planck Institute for Heart and Lung Research from a group. They were previously working with embryonic stem cell line, which is sensitive to neomycin. And normally they work with a orbital shaker, and this is actually a comparison between orbital shaker and the CERO. I don't show the data. If you're interested, I'm happy to share them later or when we talk. The bottom line is normally they add neomycin on day 8 to avoid all non-differentiated cells. When they did it in the CERO, and they see there is no effect on the neomycin, so the cells have differentiated quantitatively into cardiomyocytes, and you see also here they're very contractional, so they're very active, which is always a good sign. They here don't look so much, I mean, this is a natural homogeneity of just throwing in cells and let them do what they want to do. You can improve that, but however, these guys are going back into 2D. So their purpose is to have healthy, differentiated, mature cardiomyocytes, and then differentiate them back to 2D layer experiments for drug testing, for example. So this is actually embryonic stem cell line. There's another published data where they were comparing then the human iPSCs versus hanging drop. This group was not looking at was not looking at marker expression for cardiac marker. I just added 2 here. And on day 7 after induction of differentiation, you see in both cases it matches somehow here the differentiation, that the cardiac marker expression is improved versus hanging drop. And also you see this is this column pictures from CERO. These are pictures from matrigel. You see there's a better defined surface, and you see more organization, better organization, which probably is also a good indication for better maturity. Any questions to this point? But you didn't fall asleep, right? I didn't. Okay, good. Other examples, this is just a retina organoid, and you see very often, this is also something we see, the very nice polarity. So you see the different kind of layers that you normally would expect in a retina. And actually, I also added because I like the colors. It's just a very nice pictures, right? These are data from Australia, from a recent data. So this was actually demo data from a group in Sydney, and they were struggling usually with expansion rate of their iPSCs. They're not growing very well. You see, like 2.4 times or 2.8 times. When they did the same thing in the CERO, the factors grow up dramatically. So you see the expansion was much better and much more effective. This is one side which was nice, but what was more impressive, they were looking at the lung organoids. So driving the iPSCs into lung tissue, and you see here different markers that has been expressed, and you see they're not showing or they shared, but I didn't show here, the results from plate on day 7 or day 10. There was no positive markers for lung already on the things, but here you see the markers are where they are. You see the alveolars already forming. So quite nice result. Good. Now I would move on to tissue culture. So it's a different discipline, but as effective as the iPSC or pluripotent derived one, maybe even a bit more difficult and more labor intense. So normally what you do is you have a plate, when you're using the CERO, you have either already groups that have their tissue in a plate, let it be 24 well or 6 well, or cryopreserved. So what you do is you wash, you shredder, you disrupt, you put them in a tube. If you start with tissue, you can use a tiger and then quickly analyze it and put it into a tube. This is a standard protocol. We have, I'll tell you later, special protocols if you have very few material or very low amounts. So you put it in here, they inoculate, they form the initial aggregates, and then after a couple of days, you can split them again. So they grow quite quickly. Here you see hydrogel. So the point is, very often if the cells have been exposed to matrigel, for example, in the previous stages here with the domes, the cells like to maintain matrigel. So they get somehow, I always say, a bit addicted to it. So they grow better if you add 2 or 4% matrigel to the media. So normally what we do is we have half of the media with the cells, half of the media in cold, where you dissolve the matrigel and then you mix it together. And we have great results for stomach, intestine, pancreas, lung, liver, many cancers. And this last sentence is very important. We have later on a result where a group started with 1 well of a 24 well plate, no problem. People started with only 50,000 cells, it works. So you can, even if you have only very low amount of tissue, of cells, you have a good chance of getting it into culture. So what are those results? This is shared from us with from Dr. Eglatine Ballant from Monash University in Melbourne. And the first thing, she's a big fan of the instrument, but the first thing she was looking at is what happens if I have a few cells? What happens if I have a lot of cells? And basically, yeah, it just works. So you just inoculate. This was a standard culture in plate. You see there's not so much difference, maybe a bit more maturity. And her feedback was, yeah, it's scalable for cultivation. She can get extremely high yields and there is no compromise in quality. Very simple take-home message. Then she was looking at seeding concentrations versus matrigel concentration. So here you see the standard experiment on a plate. Here you have free-floating organoids from human breast cancer, so a bit more difficult than colon cancer, obviously. And the difference is we have 2 different seeding concentrations, 150 and 250 K, 150 or 250, and the presence of 2 and 4% matrigel. I don't want to make the story long. Nobody will see much difference here. So basically, at this stage, if you look just at the results of the incubation on day 14, it looks pretty much the same. But there are differences. And the differences start when you are looking at the alternatives later. So the time and the workload. So this is probably those that work with tissue culture know. In this case, they start with 4 wells or 5 wells of a 24 well plate. They have to passage it 4 times and they go on 15. Then they have to passage 4 times and they go on 43. They have to passage 4 times and then they're roughly around 10 million cells. And that's the purpose. And what we did, we were looking at the cost that they have for material and so on, to make a comparison, obviously. And what they found out, and we were always proud that we can save so much of the material cost, which is true. So if you look at it, we just take a tube, a CASY tube, a CERO tube, sorry. And you need much less amount of media, much less of matrigel, and plastic, okay, is a bit higher. But overall, you use roughly 65% on the material cost, which is great, right? But the major difference comes on labor. on complexity of work. And you see this is for changing and things like that. And you see this drives it down to 90%. Because, I mean, you just have to change the media 4 times, which takes you 5 minutes. So it's really low maintenance. And if you look overall, and we standardize it now on 10 million, this was 11, this was 13, you roughly have a total saving of 90% of cost, which is, I think, a huge number. And if you're doing this, I probably don't have to tell you how much money you spend on this kind of work. But that's not the only benefit. If you compare the cost, this is now in Australian dollar, you come down from 2,500 Australian dollars to roughly 250. So that's huge. But the other striking aspect was that you can drive the time to get to 10 million cells down from 6 to 10 weeks to 2 weeks. And can you imagine if you want to do like trials of different kind of treatments or things like that, that's huge. That's a big difference. So I don't want to go into much detail, but this is probably the take-home message. It's 10 times cheaper, it's 4 times faster, and it's absolutely low maintenance. And as Egler team says, it's super easy to use. So that probably is the take-home message. If you want to improve, if you want to spend your resources where it makes more sense than just on routine work, and save some costs with it, then probably this is the best solution you can find. But it doesn't stop there. Now we have shown you it's easier to get there. It's probably more mature. It's probably more vital, and that. But what else can you do? And I just have one example in here, which is from a group in Germany in this case, and they are looking for co-culturing. So they want to develop brain organoids and co-culture them. So let's first look at the brain organoids themselves. Here you see the situation that they were normally having using an orbital shaker after 3 months. If you work with it, it's probably a familiar picture. They're very heterogeneous. You have this very light center, which correlates with a lot of apoptosis going on. And if they're growing, they're only growing on the surface levels. So that is probably what you were familiar with. After a certain amount of time and after a certain amount of size, and which limits you in your experiments. As opposed here in the CERO after 5 months, so 2 months longer, you see the density in the middle is much better. It's proliferating in the middle and in the outside, so through and through. And yes, there's only a little bit of apoptosis in the middle. They're evenly sized, and I don't want to go that it's much higher capacity and so on and so on. So a lot of benefits here. I don't want to go into, but just keep that picture as I think explaining the benefits by himself. So the next thing they were trying is then to go into co-culture. And this is just one example of what is done. So they were using a glioma spheroid, and they were taking the cerebral organoid. They put them in a U-shaped well plate to keep them into contact. And after 72 hours, they put half of the wells into the CERO, and the rest of the culture remained in the plate. And then after another 48 hours, so 2 days in the CERO, they were analyzing the data, and you see, and that was actually quite impressive for them, that the fusion, the tumor invasion into the healthy tissue has progressed much more. Currently, they're publishing the data, so we have this preprint where they can show that myeloma to brain or other tissues to brain, they really have migrated into the healthy tissue. And if you're interested, we can share the publications and the data. And as a tumor model for treatments, like, sorry, I'm using simple words, that you kill the right stuff and not everything. So basically. As opposed here on the plate, you have very poor interaction, and normally they stop very early because you see apoptosis already coming up. So they're about to die anyway. Good. Some other stuff unrelated to cancer. This is healthy tissue. It's stomach. It's done from one of the best German universities. It's from Technical University of Munich. And they're looking at Helicobacter infections. So they need stomach organoids, human stomach. And this is a typical organoid on day 22. So you see they're single layer cells, heavily folded, high complexity. But you see it's really stomach, it's not colon. And these guys are also doing inside out for the study. So that's also what you should keep in mind. You can have your organoids, like stomach organoids, normal in a normal folding or inside out. So it's both possible. What they also said, like they start, and this is something what we see very often, people start with one instrument and after a year or so they add one or two more. And that is always for us a good sign because the platform works and they're quite happy. And they save a lot of money, which you then can reinvest for more instruments because they have also more time to do more experiments. These are just some data on liver. I'm going to skip this maybe. Yeah. And coming to the last slide, this is, as I said, more a niche where people are taking dissections of liver pieces and maintain them in the CERO. So this was the first experiment that was done still with phenol red. Normally, liver people don't like phenol red, so then there was a later culture where they were going then up to, and they studied like LDH concentration, ADP, and a lot of markers that go along. I'm not familiar with all of them, but they're looking at different factors to see how the liver is maintained there, and they came up to like 21 to 27 days, which is enormous. It has been done for other tissues as well. So it can also be a way if you get the tissue late in the evening, you cannot process it anymore, you want to do it the next morning. So it's a good way of storing them overnight and without losing anything. That brings me to the end, and I hope you agree. The system can provide very easy handling. So as Eglantine said, super easy to use. It's also a standardization. So we heard a lot from pharma companies that we are working with that they can finally standardize their work. It improves the viability, vitality, maturation, polarity, so a lot of highlights here. It gives you high yields in a very small volume. Just remember up to 80,000 iPSC-derived kidneys in 40 ml. You can start with very low volume. We have some special protocols also for tissue pieces if you just have very small amounts to scale it up and then to go into a routine process. You can do long-term very often, and you have a lot of cost savings with it. And that's it. Thank you very much. Questions?
Hi, Joachim.
Hi.
Hi, Joachim. Thanks for the great talk. It was very informative and useful for us. I just have a general question about automation in this system. So, what is the current level of software compatibility or physical integration standards to connect the OLS instruments to third-party robotic handling platforms? Is there any ideas in that, any limitations that we can be expecting?
Yes and no. I think we're working on an update of the model, which is probably coming next year somewhere. And then probably the interaction is much, much easier.
Okay, I look forward to that.
Yeah.
Thank you, Joachim.
No problem. Other questions? I mean, normally I would ask, like, what are you working on before we start the talk, but I think that was a bit limited today. So let me know if you're working or is there any more detail you want. You have my email address. No, you don't have it, but you can send it, like, I'll go back to the beginning, and you can send me also an email if you want and have more information. It's down here. And yeah, other questions? Okay, that doesn't seem to be the case. So think about it. Oh, sorry. There was someone?
Hello.
Yeah, this is Agnes from Gaia Science.
Yeah. Agnes.
Hello, can you guys hear me?
Say again?
Hello, can you guys hear me?
Yeah.
Yep. So, is there any other questions or concerns from the floor? If not, before wrapping up, I would like to first thank you, Dr. Joachim, for providing this insightful and informative presentation today. And also a big thanks to A-Star MCB Daniel for co-organizing this webinar with us. And of course, to all of you who joining and participating this webinar. So we also excited to share that Gaia Science Singapore will be receiving a demo unit for the CERO 3D incubator for all the researchers and come on to have a try. So please feel free to reach out to us if you have any questions or if you're interested in this to getting know more or we can discuss how it can fit into your research flow. Yep, so we are the distributor in Singapore here with partnering with OLS. So thank you everyone for attending.
Yeah, thank you very much for organizing, and have a great Friday, a great weekend in front of you, and don't hesitate to ask either Gaia or us if you want to have a trial. They're very easy to process protocols, and believe me, whatever you do, not whatever you do, but if it's somewhere related to what I just showed, it's very easy to translate to work in the CERO, and you will see the improvement within short. Thank you very much, and then I drop off. See you.
Thank you everyone.
Thank you.